Hello everyone, good evening. Thank you all for joining. This is the second problem solving session of the course Tissue Engineering. Those of you who have joined this week for the first time, let me briefly introduce myself. I am Mo Chandra. I am a PhD scholar at the Department of Chemical Engineering at IIT Kanpur. And for my thesis, I am working in the field of tissue engineering. During the course of my work, I have gained experiences in various biofabrication technique with various kind of biomaterials. If you like to know more about my work, there's my website. You guys can go and check it out. If you have any course related queries, you can ask me during the sessions, or you can drop queries in the course discussion forum as well. So every week, Saturdays, we will meet like this from 4 to 6 p.m. You guys have the link with you. The session is recorded so that it will be uploaded in my YouTube channel. You can watch it later in your own time. The notes that I'll use during the session is also made available, and attendance will be recorded, all right, for the sessions that you're attending. And as well as towards the end, I plan to take one revision session as well before your exams. All right. So the platform that you guys joined through is MS Teams, and you may not be able to see the chat option. All right. So if you have any queries, you just have to raise your hand, or you can directly unmute yourself and speak. If you do not have any queries, I request you to mute yourself so that noise or disturbance doesn't happen, all right? And we can conduct this session smoothly. So let's begin. We will, like in the slide, I'll just briefly recap what you have read or watched in week 2 content. So, as you all know, tissue engineering or the field of tissue engineering talks about three things, right? The biomaterials that is going to be used to make the scaffold, the cells which are going to be seeded on scaffold or combined with your scaffold in some other way, and then there is signals. You deliver some signaling molecules to direct the behavior of cells. So this is called the tissue engineering triad. So in week 2, we talked about the first triad, first member of this triad, that is biomaterials. All right. So we looked at what type of biomaterials are used, like there are natural polymers are used, synthetic polymers, then decellularized ECM is also used, and these biomaterials are used to make a scaffold. So why we are interested to make a scaffold? Scaffold basically mimics the ECM, extracellular matrix of our tissue, and it will do similar function that the ECM does in our body. The primary function that you can all think of is to provide mechanical support for cell growth, because you know mammalian cells are adhesive in nature. They need a hard surface onto which they can adhere and then do the process like differentiation, proliferation, etc. So you need to provide a surface. That is the primary job that the scaffold is supposed to do. Other than that, there are other properties as well that makes it suitable for a particular tissue engineering application. And what kind of materials that can be used to make scaffold? It can be from natural origin, it can be synthetically prepared in lab, or you directly take the tissue, remove the cells, and you get intact decellularized ECM. All right, so that itself can be used again for culturing another type of cell for a different type of tissue. So you take these materials, so these are the biomaterials now, you now fabricate scaffold out of it. So how you do it? There are various methods available. You can 3D print it, you can electrospin it, right? Or you can use lyophilization technique, or you can use solvent casting, porogel leaching technique, and like you can make beads, micro beads kind of structures. So various type of methods are available, but the choice of method, the choice of the polymer, to fabricate the scaffold, the geometry of the scaffold, it all depends on the type of tissue that you're targeting. If you are concerned about making an artificial bone, then the properties will be different from that of a artificial, say, cornea, right? So uh, then the properties of the materials, method of fabrication, all the things will change. So whenever we start thinking about tissue engineering, or whenever we start a project in tissue engineering, we first have to understand what organ we are targeting and the properties of that particular organ or tissue. And based on those properties, we select our biomaterial to fabricate scaffold. We select the technique to uh, fabricate the scaffold, and then the cells will come, what type of cells you will use, then which signaling pathway you want to target for a particular effect. So if your purpose is to create an artificial bone that can replace lost bone, right, in the body, so then the properties should differ. If you only want like regeneration of bone, then the scaffold properties and the material that you will use will be different. So depending on the purpose, what purpose you have thought in the beginning of the project, other things like the triads of tissue engineering will be selected. And lastly, we also saw hydrogels. All right. So hydrogels are nothing but a form of a say natural polymer, synthetic polymer. All right. Form. All right. It's a physical form with a unique ability to absorb large amount of water. Okay, so usage of hydrogels is very common in tissue engineering because of the unique feature that it can create an aqueous environment and mimic what is there in our body. We are mostly water, right? The cells, all the growth factors molecules, so they are all present in aqueous environment. So when we want to mimic that, the obvious choice of material or type of material is hydrogels. Other than that, people also use fibrous materials, like they create fibers out of it using different techniques. All right. So we just started the course, and now now is the very nice time, to explore and understand what everything each of these means. Because later on the course, when you will see that two, three components are used together, then the things become more complex. So if you have any doubts for week one or week two content, please feel free to ask. Also, we will briefly discuss in detail some of the topics of week two through questions. So during that also, if you feel like I haven't covered some part, all right, and you have query on that part, you just unmute yourself, you can speak. If you feel shy, you can raise your hand, I'll call upon you, like whatever is comfortable. Only thing is chat option is not activated here. All right, so you have to speak, you have to interact in order to get your queries answered. All right, so now we will begin with the questions. So till now, anything anyone wants to say or has any comments or questions regarding anything?
Excuse me, can I?
Ah, yes, yes, yes.
Uh, can you exactly explain in simple terms what a scaffold is and what a chondrocyte is? Am I pronouncing it right? Chondrocyte.
Chondrocyte.
Yeah.
So let's talk about chondrocyte first. So chondrocyte is the resident cell of the tissue cartilage. All right. And when we talk about cartilage, the cartilage itself is a very unique type of tissue and very easy to engineer in comparison to others. Why? Because it is mostly collagen or mostly ECM, whereas chondrocyte is very less in comparison to the ECM content. Also, uh, for the growth of chondrocyte, it doesn't need a network of blood vessels or vascularization. All right. So that is why cartilage is avascular, and avascular tissues are easier to engineer in lab. Other than that, chondrocyte have other functions as well, like any normal cell does, like they can proliferate, they can migrate, right? They can adhere. Other type of these functions is possible. Another important thing is it is of mesenchymal origin. So during development, our cells, the few number of cells that are there in the beginning, they divide, all right, and two lineages takes place. One is epithelial, another is mesenchymal, all right. So most of the connective tissues like bone, cartilage, then blood, so these are all some examples of connective tissue. They come from the mesenchymal origin, whereas the epithelial cells like our keratinocyte cells, intestinal epithelial lining. So these come from the epithelial origin. So, like, I hope I didn't make it complex for you to understand chondrocyte. But the thing is, chondrocyte is just in one line. If I say it is just a resident cell of cartilage. Then you also asked what is a scaffold. So scaffold is a general term. If you Google it, you will see that scaffold has different meaning in different fields. So when we talk about scaffolds in tissue engineering, it is nothing but artificially we are trying to create the ECM, extracellular matrix. Artificially we are trying to create. Why? Because the cells, the mammalian cells, they need a support structure onto which they can adhere and grow. That is why scaffold is needed. If you just use a cell suspension in a liquid, in a media, cell culture media, and try to deliver it, suppose into your body, then first is cell loss will be there. Adequate cells will not reach at the target site. Okay. And the cells that you're delivering, they will have a less propensity to grow, means they will not, they will find it very difficult to grow. Whereas if you deliver your cells with a scaffold, the scaffold itself is providing the initial boost or kick to the cells to grow. All right. And when it reaches or when you transport it to the site, then the effect will be faster. So if you're looking for faster wound healing, faster regeneration, so it will happen faster if you use a scaffold. All right. So now depending on what tissue you're targeting, the properties of the scaffold might change. Some unique properties you will have to add, like if you're targeting neural tissue engineering, then some conductive properties have to be added. Like if you're targeting corneal tissue engineering, then optical transparency we are looking for. So depending on what tissue you're targeting, accordingly you will choose how you want to make your scaffold. I hope my explanation was clear.
Yes, thank you. Thanks.
All right. So if there is no further questions, we will proceed now with problems. All right, very simple problems. The problems which are related to your week 2 assignment, but I will not discuss the assignment. All right. So you can ask me doubts related to your assignment, but I will not tell you answers. Please note that. Okay. So the first question related to the what is scaffold. So this is the question. Choose the right option about the given statement. Scaffolds are a type of biomaterial that help in support and delivering cells. They have application in tissue engineering, drug delivery, and regenerative medicine. So is this statement true and false? It's true or false? Any one is correct. speak up. I expect the answers from you.
True.
If any, true. All right. So you are correct. I guess Gargi spoke. You are correct. It is true. So it's very self-explanatory. I just explained what is scaffold and what is the function of a scaffold. So related to that only this question is all about. Okay. Then now moving on to the next question. Okay. Which of the following is not a desirable property of a scaffold? So when we're talking about properties of scaffold for this particular question, think about the general property that a scaffold should possess irrespective of the tissue type. Okay, we are not talking about tissue specific properties, rather we are talking about the properties that generally a scaffold should have, no matter for what application, what tissue engineering application you are using. Okay, so here are the options. It should be non-immunogenic. It should have high volume to surface area. It should contain cell adhesion sites, and it should support cellular fate processes. Now, these are four options. You have to choose which is not desirable.
Option B, high volume to surface area.
Okay, so you're correct. I know you just answered. I also want to see what others think. Do they think that B is correct? Yes, go ahead. Are
Ma'am,
you
can you please explain what is cellular fate processes?
I'm really sorry, but I couldn't understand what you just
Hello,
said. Yes, now your voice is clear.
am I audible?
Yes, yes, now you're audible. Yes, go ahead.
Can you please explain what is cellular fate processes?
Okay. So, cellular fate processes is a general term or is a means term collectively used to explain the processes that usually any cell type goes through, like cell adhesion, cell migration, cell apoptosis or cell death. Rather, good term should be cell death. Then cell proliferation, cell differentiation. So I'm trying to say that a scaffold should, like, it is not correct. I don't know. I'm not telling you the answer, but I'm trying to say that when we want to address all these processes together, we call it cellular fate processes. And any cell type in our body will go through all of these processes. Cell adhesion is not there for blood cells. Other cell types in our body do adhere on surfaces. All right. So there is one exception. Other than that, all cells will migrate, they will die at some point, they will proliferate, they will differentiate. Okay. So as I was saying, yeah. So as I was saying that one of you have answered that option B is correct. What about others? What is the correct answer according to others?
I feel it is B, like the
It
same
is
as
B.
you said.
Okay, it is B. So both of you are correct. It is B. We want rather high surface area to volume ratio. Why? Because cells do not see bulk. Cells see the surface. So if the surface area is high, there is more space for the cells to adhere. All right, and um, adhere, and then it will proliferate, and then it will differentiate, migrate. Means more space. Basically, we are trying to give more space to cells. That is why we prefer a porous scaffolds, where the porosity is such that it's like interconnected, like tunnels. Tunnels are there in between adjacent ports through which not only cells can migrate but also uh, continuous flow of nutrients, oxygens, etc. is also possible. All right. This is the next question. What is the expected response of scaffolds when it is transplanted in vivo? It should initiate bacterial infection at the transplanted site, promote tissue reorganization, remodeling at the transplanted site, promote chronic immune response at the transplanted site, all of the above. Option B. Okay. What about others?
Option B.
I think it's option C.
Option C. Okay. So we got two answers, B and C. So the correct answer is actually option B. Uh, when we are say making a tissue engineered product where the scaffold is seeded with cells and you have cultured it for some time and now it is ready for transplantation. So your product has both the cells and scaffold together and it is transplanted. What we want is it should, like if you're trying to address the tissue repair problem, like some wound has happened and you're trying to repair it, then the expected response that your product should promote healing and then reorganization. More cellular growth should happen, more ESM generation should happen, and then the injured site should almost go back to its original state. But if your product is not sterilized properly, then it can contain even one bacterial cell or few bacterial cells that can cause bacterial infection at the site. So rather than doing benefit, you are going to do harm. And if the site is injured, like doesn't have proper exposed to a body, like in burn wounds, the site is exposed to environment, right? So in those cases, you are making the trouble much more 2x times you're increasing. Similarly, any foreign body, for option C, I'm talking any foreign body that you implant inside your body will generate some immune response, no matter how biocompatible it is. Okay, even if you are not providing any substance that you know is not going to provide an immune response, still some level of immune response will be there. So we want to minimize it. We want to make sure the immune response should be as minimal as possible. It should be of acute nature, not chronic. If it is chronic, means all the like inflammatory factors are there at the site for a longer period of time. Then what happens at the end? That rather the purpose of tissue remodeling is not happening. Rather, a new problems get created that has to be removed surgically afterwards. So, ideally, what is expected from a tissue engineered product is that you place it at your injured site. It should integrate well with your host environment, okay, host body's environment. It should allow infiltration of the host cells, growth of host cells onto it, secretion of natural ECM, and then over a period of time the biomaterial if you have used it will biodegrade at its own rate, and ultimately what you will see at the site is almost original state of the natural tissue. Means secondary surgical intervention is not required at all. If here in this question A and C, if suppose happens both of these is going to recreate a need for intervention. All right. So that is why option B is correct. Okay. So one of you have answered C. Did you get it? Why C is not correct?
Yes.
Yeah, great. Now this is the next question. Which is the most abundant protein in the body? Collagen, keratin, hemoglobin, immunoglobulin. The question is for you guys to answer. If anyone knows, you can unmute yourself. Collagen. Okay, be confident. Why it's collagen? You should say collagen. Okay. All right. Anyone else would like to try? Yes, go ahead. It's a simple GK types question.
Collagen.
Collagen. Okay. So we got two response. So correct answer is in fact collagen. And in fact, collagen makes 30% of our total body's weight. All right. And among the total collagen, 90% of it is type 1 collagen. Whereas you see keratin in skin, in our hair, in our nails. So it's a protein responsible for like protecting us, like structural protein. It is a kind of structural protein. Then hemoglobin you see in blood in RBCs, responsible for carrying blood. Immunoglobulin are basically the antibodies help in immune responses. All right. So now again, based on collagen, which of the following amino acids are predominant in collagen? Okay. Option B. Anyone else?
Option A.
Option A. Yes, I got an answer. Option B. Anyone else have anything else like option B? Right, right. I marked it.
B.
Doesn't. Yes, option B.
Yes.
Option B. Okay. So actually, the correct answer is option A. Okay. Why? Let me show you. So this is a chart that I took from just Googling the amino acid distribution in collagen from different sources like rat, bovine, codfish. And if you see this table carefully, and this is the weight percent of different amino acids in, some total weight of collagen. So if you see here, glycine, then hydroxyproline, then there is proline. If you see the percentages, it is much higher with respect to other amino acids. I'll give you 2 seconds to observe, to make the same observation as me, and let me know if I'm wrong. Whereas lysine here, have a look, is comparatively in much less weight percent. All right. In fact, I read somewhere that these three amino acids, glycine, proline, and hydroxyproline, makes 50% of the total amino acid content in collagen. Rest 50% are the remaining amino acids in this list. Okay. Did you get why glycine, proline, and hydroxyproline are the predominant ones in the collagen?
Yes, ma'am.
Yeah, great. Now, this is the next question. It's a simple match the following. Whoever is able to get even one, you can unmute yourself and speak up. This question is straight from the lectures. What about A? Option A. It matches correctly with. Yes, anyone?
A.
I can go
Structural
with both.
proteins.
Okay, Rijuna, you speak first, then Devika, you can answer.
Nucleotides, gas, liposomal, first one. For B, it is one.
Um, your voice is very low. Like, can you speak up? It's very difficult to understand what you're
For
saying.
D, for D, it is glycosaminoglycans.
So you're saying D, proteoglycans, is one, correct?
Yes.
Okay. Now, Devika, you can go ahead.
A. Fibronectin laminin, specialized proteins. Collagen type and elastin fibrillin, it is structural proteins.
Okay.
Then hyaluronic acid heparin is glycosaminoglycans. Proteoglycans is the first one.
Okay.
Glycosaminoglycans belongs to protein.
Yes, yes. Great. Thank you. So, now, great, we matched it. Now, I would like to understand, like, it's good that you memorized, but what logic would you have, like, did you have in your mind while answering? Okay. So, first simple question. Why fibronectin is classified under specialized proteins, or why laminin is classified under specialized protein? Anyone? What's so
Because they
special
have
about it?
other binding sites and also they are specialized. So it can bind to cells, it can bind to other collagen, so it can
Okay.
bind to heparin. So they are specialized proteins. While
Mm,
collagen type 1, they are structural proteins because they are very structured based on the fibrils it has structural proteins.
okay. So, by the way, collagen also has the same cell adhesion sites, right? Similar cell adhesion sites. And yeah, anything more, like anything more that you can recall about fibronectin or laminin that makes it specialized? Yeah.
What is the cell for matrix?
Yes. Can you speak up?
What is the cell for matrix?
Yeah, that is a component of ECM. Yeah. Anyone else would like to try? Why it is specialized? What's so special about it? So if you recall the structure, like from the lectures, if you recall the structures of these two proteins, you will see other than cell binding sites, there were other sites too, right? There were heparin binding sites. Then there were uh, sites for different kind of cell receptors to bind. Like there is a cell receptor called hemidesmosomes, desmosomes. So those kind of receptors can also interact with these kind of proteins, and they itself has like sites to bind to growth factor molecules, so that a local distribution of growth factors can be created. And so yeah, that is why it has additional functions. That's it. Whereas elastin, fibrin, collagen, their structure, if you look at it, collagen is triple alpha helix. So it's a fibrous structure. So that is why it provides strength, and a fibrous matrix is created. Elastin or fibrin itself, both of them together, they interact and gives elasticity to certain type of tissues like skin, right? So they predominantly contribute in deciding the structural properties of a tissue. Okay, the predominant, if you remove the collagen from skin, you won't be able to get complete property of skin out of it. Means like similarly to skin, bone, right? So bone, the main function of bone is what in our body? To allow movement, right? That is why they are much more uh, stronger. It can resist compressive and tensile forces. So from where that property will come? It is coming from the presence of collagen. That is why it is called structural protein. So you have to relate like that. Means what tissue related function it is doing, that particular protein, how it is helping tissue as a whole, organ as a whole to function. Okay, then it makes sense. Then it makes sense why it is classified like that. Okay, so apart from proteins, you saw in ECM there are polysaccharides as well. So the polysaccharides which are present in tissues are called GAGs, or glycosaminoglycans. So the example here is given as HA and heparin. So the main function of GAGs, anyone, what gags do in a tissue, why gags are needed?
Impact viscosity.
Viscosity, okay, fine. But anyone would like to like say it in much more better way? Viscosity is fine, but it's very vague, so bit more specific.
Um, the, the,
Anything?
the, the gangs
Yes.
tend to improve fluidity because of the cross-linking.
I'm really sorry, but I couldn't understand what you said. If anyone else understood, can you speak in behalf of him?
Now,
As a lubricants,
Lubricants, okay, right, makes sense. So correct way of saying is gags are also present at locations where lubrication is not needed. Means in normal skin type of tissue where you don't need a lubricant, right? To bear the load. So gags help in giving the ECM a fluidic nature. It is it makes the ECM gel like, so that it can absorb a large amount of water. Okay, so and we try to mimic it. As I told you, we try to mimic that property of ECM through using hydrogels so that it can hold a large amount of water. It is similar to gags. So protein, if you see, they have some specialized functions, they give strength and all, where gags in its own is related to making the environment much more fluidic, so that the cells can freely migrate, the molecules they can freely diffuse through a matrix, and it should stay confined. It should not diffuse at a rate like it diffuse in water, but it should diffuse at a slower rate so that the cells can sense its presence, right? So in that kind of environment, gel like environment, gags help to provide. So that's why if you are using gags at a higher concentration, or gag like material at higher concentration, then the viscosity increases, right? Like glycerol, it's a very high viscosity. The similar viscosity you can get. Lubrication, yes, definitely, it's fluidic, so it can provide lubrication. And proteoglycans remains, so it's like a hybrid kind of a material to my understanding. Means it's a gag molecule linked to a protein core. Okay, so it's trying to bridge the differences between the proteins and the polysaccharides, and like interacting with both the components so that it makes up the overall properties of a tissue. Okay, so I hope it makes sense, and it will help you better understand. This kind of understanding will help you better understand why each of these components are needed in an ECM. So in future, if you want to create a tissue artificially, you should know like what properties or what material should I add so that this property can be mimicked of that is coming from this particular ECM component. Okay. So far, whatever we have discussed, any queries so far? Okay, I'll assume that there are no queries. Moving on, among the following methods, which one of the following is used to desolarize tissues? Freezing and thawing, trypsinization, using detergents like Triton X, all of the above.
And B.
D. You said D, right? Or B?
B. B.
B. You said B, trypsinization?
Yes,
Or
I'm
o- okay, I'll mark B for you. Anyone else would like to try? Yes, sorry, I didn't get it. Can you repeat? So one of you have answered B. What about others?
all of the above.
Yes. Can you repeat?
All of the above.
All of the above. Okay. Okay. Anyone have anything other than these two options in their mind? Does anyone think it's A or anyone thinks it's C? Okay. So I'll assume that most of you guys think it's either B or D. Okay. So let's see what's the correct answer. The correct answer is D. In fact, all the three options are used one of the methods to desolarize a tissue. What is desolarization? Anyone?
Removal of cellular components from the tissue.
Sorry, I couldn't understand. Can you repeat? Maybe slower this time.
Removal of cellular components from the tissue.
Yes. Did you say cell removal, something like that? Yeah.
Yes.
Yeah.
Removal of
Okay.
cellular components.
Okay. Um, okay, fine. So you're correct. So we are trying to just remove the cellular component from a native tissue. We take either tissue from human origin or any animal like bovine, porcine, etc. And then we remove the cells. And we have to do it in such a way that any cell related component like cell debris, cell membrane components, cell proteins, um, nucleic acids, like everything should be removed. And why we are focused on removing the cellular components? Anyone? Why do we need to decellularize it?
To avoid the effects of immunogenicity.
Immunogenic response, yeah. So we cells or the proteins that are present on cells, like we discussed in the last class, most of the proteins that contribute to immunogenic response are the IHC complexes. So they are like easily recognized by our body's immune cells, and once it's recognized, then very extensive, very harsh immune response will result. In order to prevent that, and allow the tissue engineered product to get accepted by your body, we want to remove the cells. So while removing the cells, you have to make sure that the surrounding ECM is intact. You can remove cells as harshly as possible, but if the ECM is not intact, if the ECM properties are changing because of your method, then it's of no use, because ultimately you are going to use that ECM or decellularized ECM for making a scaffold, right? And onto which you will sit another type of cells and make your tissue product. So among these methods, so first method, the freeze thawing method, is in fact is a physical method where we do not use any chemicals here. We just use the phenomena of freezing and thawing. We keep it in lower temperature and then thaw it. So what this does, it will cause formation of ice inside the cells, and cells will rupture. Similarly, we can sonicate it. We can use a sonication bath where the cell's membrane will rupture. These are physical methods. You are physically trying to kill the cells, lyse the cells. Then there is enzymatic methods available where you use enzymes like trypsin, and you like basically break the bonds between the cell and your ECM. So what trypsin does, it uh, like acts on the cell surface receptors like integrins that bind the cell on the surface. So because of which the ECM is not uh, harmed in any way. The ECM component is not at all harmed. In fact, we are just gently removing the cells. We are just stopping the binding and just gently removing the cells. The cells are removed intact in this case. So it's a very gentle process. And we use it very frequently in our cell culture practices where we uh, trypsinize, or we add the enzyme trypsin. We remove the cells from one vessel and we transfer the cells intact, live, to another vessel. Okay. Then we can also use chemicals like detergent. Tritenex is a very commonly used detergent where these detergent molecules, they basically rupture the cells, all right, and wash off, and whatever byproduct that you get is like decellularized. That's it. It's a harsh process, so you have to optimize how much detergent to use. If you use very high amounts, then you can also structurally break down your ECM. Okay, so you have to be very gentle in your approach. Then you can also use mechanical methods, which are by methods like the peeling off or brushing off with some mechanical tool. Itself sounds very harsh. So again, you have to be very gentle as much as possible without corroding or damaging your ECM. You have to remove the cells. So generally, in our lab, we have decellularized plant cells, plant tissues basically, and use the decellularized plant or leaf, you can say, to culture the cells. In those aspects, since plant cells, they have cell wall as well, so that are much more stiffer. So we have used Tritenex in our approach. But other type of substrates like animal origin, we better, I think, for me, I would go with trypsinization. It's very gentle. The also the animal ECM is much more what do you say, is soft, all right. It's very, it have to be handled with care in comparison to plant ECM. So, so these are the methods that are reported, and detailed protocols are available over the internet. People routinely use it to create decellularized ECM and then use for tissue engineering applications. Okay. Now, in the same track, I have another question for you guys. Which of the following is a commercial decellularized ECM product? So these are the examples. If you recall, then it's very straightforward to answer.
All of the above.
Option, can you repeat option? Which option did you say?
Option D.
Option B or D?
D. All of the above.
All of the above. Okay. So yeah, you're correct. It is all of the above. If you go back to your lectures, you will see that professor have discussed the origin of each of these commercial product that is available now, from where it is coming, from which tissue type is used to create it. So it is in detail it is explained in the lecture. So I will not go in detail here. It's readily available with you. Okay. Now, which are the major components of ECM? Again, a very simple question based on the discussions we already had.
Option C.
Option C. Yes. So it's correct. It's option C. Again, I don't need to go into much detail. It's pretty self-explanatory. Now, mm, coming to next question. Yeah. Which of the following?
Previous option.
Yeah. You want
So, previous slide.
previous slide. Okay. So I have changed it. So give few seconds. Yeah. This is this previous slide.
Yeah, okay. Fine.
Yes, you have a doubt here?
No, ma'am, no doubt.
Okay. All right. So now can I move to the next question?
Yes, ma'am, sure.
Yeah. Yeah. Okay. So this is the next question. Which of the following has both type of glycosidic bonds 1,4,1,6? So for this question, I can like give you freedom to Google the structures. Okay, don't search it. You can Google the structures of cellulose, starch, silk fiber, and keratin and answer. But don't search the answer.
Starch,
Be honest.
starch.
Yeah, it's starch. Okay. Why is starch?
It has both 1,4 and 1,6 like acidic.
Okay. Glucose, sorry, cellulose it doesn't have
Has 1,4.
1,4. Okay. Now can C and D have this glycosidic bonds?
Keratin doesn't have. Silk fibronin, I don't have. I don't know.
Actually, glycosidic bonds are only there in polysaccharides. All right. So C and D are they are proteins. They will have amino acids and they will be peptide bonds. So C and D is like out of question. They cannot have glycosidic bonds. Okay. So it is correct. Starch have both 1,4 and 1,6. In fact, starch have alpha type of glycosidic bonds, whereas cellulose it has beta 1,4 glycosidic bonds. Okay. So if you are not sure how the glycosidic bonds look like, just simple Google it. It's a school standard question. Okay. And this is the next question I have. Which of the following has only beta 1,4 glycosidic bond? There are multiple options that can be correct. Yes, go ahead. All of these are polysaccharides. I will not fool you like last time.
Cellulose,
Um, can you repeat? Both of you can repeat. You spoke so quickly.
cellulose,
Cellulose. Okay. Okay. I got cellulose, but more than one is correct here.
dextran,
So dextran. Did you say dextran?
no,
Huh? Can you
yes,
confirm?
dextran,
Did dextran? Okay, fine. What about others? What about HA, hyaluronic acid? Does anyone think that it might be one of the correct answers? Or chitosan? You can simply Google the structures. Okay, you have the freedom to do it today, but later on, don't do it during the exams. Okay, but don't see the answer by don't Google directly. What about chitosan? What about hyaluronic acid? Okay, I'm still waiting.
Cytosan have beta 1 polymers,
Okay,
while
chitosan.
hyaluronic acid has beta 1,3 as well as beta 1,4.
Okay, so chitosan is incorrect. Sorry, chitosan is correct. And sorry, hyaluronic acid is incorrect. Sorry. So hyaluronic acid has two types of glycosidic bonds which are alternating in nature. The correct answers are cellulose and chitosan. If you see the structure of cellulose and chitosan, you will see that chitosan and cellulose both have beta 1,4 glycosidic bonds. Their cellulose glucose molecules are connected in beta 1,4, and in chitosan, I don't remember the monomers, but you can check it out. Whereas dextran has alpha 1,6, not beta. And hyaluronic acid, as noted, it has two types of beta bonds, beta 1,4 and beta 1,3, not 1,6. Beta 1,4 and 1,3 glycosidic bonds. Okay, you can memorize the monomers if you want, but bonds and monomers together, if you remember, you will remember the structure much more better. Okay, now this is the next question. Chitosan is obtained by, like, how do you derive chitosan? By alkylation of chitin, acetylation of chitin, partial deacetylation of chitin, complete deacetylation of chitin.
When it's complete desaturation of country.
Okay. What about others? Do you guys agree with her? What about others? Do you think D is correct? How do you make chitosan from chitin? Or did you understand from the lectures? Do you think alkylation is done?
Yes, ma'am.
Alkylation. Do you
Yes, ma'am.
like no, right?
Yes.
Yeah. So it's wrong. So acetylation or deacetylation, like something, any of these sort. But what about is it done if suppose we are doing deacetylation, does complete deacetylation will give you chitin, or will complete deacetylation will of chitin will give you chitosan? Sorry, yeah. What is chitosan? How would you define chitosan? Is it crystalline in nature? Semi-crystalline, right? So if you remember, the degree of deacetylation, it will um, affect the crystallinity as well. If you do 100% deacetylation or 0% deacetylation, both of this will result in crystalline structures. So in order to get a semi-crystalline nature of chitosan, we prefer doing partial deacetylation. We like stay in between 0 to 100, like 50-60%, and depending on how much crystallization, so deacetylation is done, your degradation properties and etc will be affected, and accordingly solubility in water, whether it is not, like all these properties will get affected. But either extremes we don't do, then the property of chitosan, the true property of chitosan will not come out. Okay, so we do partial deacetylation of chitin uh, to get the chitosan, and it is very, very much used like in different tissue engineering applications, like loads of literature is there. Okay, yeah, this is the next question. Which of the following is a non-biodegradable natural polymer? And you have to answer why. All of these are natural polymers, but which one of these is non-biodegradable?
Um,
Yeah,
it is cellulose because we don't have the hydrolysis that can break it down.
sorry, like some background noise is there from your end.
Like we don't have the hydrolysis, the enzyme that can break that cellulose down.
Give me 1 second and I'll use. Do not understand. Can you speak up now? I have connected my earphone. Yeah, can you speak up?
Like we don't have the hydrolysis that can break down the cellulose.
I'm really sorry, but I'm not getting. If anyone of to other than him like getting what he's trying to say.
Ma'am, is it D? Dextrin?
Okay, dextrin. Okay, I
Cellulose,
don't know
cellulose,
the
ma'am.
correct. Okay,
Cellulose, ma'am.
okay, okay. So why cellulose or why dextrin? If you make a tissue engineered product out of cellulose or dextrin, why it will not biodegrade in your body?
To digest cellulose, we need the enzyme cellulase, but we don't have cellulase enzyme.
Yes, we don't have cellulase enzyme. The correct answer is cellulose. And we are not like the herbivorous animals, so we do not have cellulase enzyme, so we cannot digest it. But cellulose as a material is a very good natural polymer. In fact, it has very good mechanical strength. So if your purpose is not to create a biodegradable tissue engineered product, then you cannot use cellulose. But if you are just trying to create structural support through your product, like you're trying to give some structural support, like in case of bone and all, then cellulose can be used. So, does alginate or chitosan or dextran biodegrades? Yes, obviously it degrades, but I want to know why or how it degrades in our body. That is also you guys should know. Why alginate biodegrades or how it biodegrades. Chitosan, how it biodegrades in your body. Then dextran. Anyone?
And porosity is much more than cellulose. That's why.
So yeah, porosity can play a role in um, also uh, directly porosity doesn't affect the biodegradability. It is the material's chemical properties and how those chemical integrity is affected. That is how it will have a tendency to biodegrade or break down into smaller pieces. Biodegradation means what? Means in presence of some maybe physiological environment in your body or in presence of some enzymes or some cellular activities means whatever activity that is happening in your body, that activity is trying to break down the polymer into smaller and smaller pieces so that it can be excreted out either through excretory pathways in your body. That is called biodegradation. Okay, so we have to think in a way that what, how, or what chemicals are there in your body, what enzymes are there, or whether the physiological environment, the pH, the temperature, all these things, does it contribute in breakdown or does it contribute in like degradation? So porosity indirectly it can, if you make a very porous structure, means you're basically creating more and more void space, then you're allowing the degrading agent to reach your site faster. That's it. But what is that agent? That is the question. So when you're using alginate based scaffold and you're implanting it and you made it very porous, then why alginate will biodegrade in your body? Similarly, if it is made of chitosan or if it is made of dextran, why it will biodegrade? All right. So let me answer this for you. So alginate uh, itself, if you are using a very high molecular weight alginate, then the degradation capability decreases. But if you are using alginate of small molecular weight, then degradation will happen through hydrolysis and but the extrusion rate means how quickly it comes out of your body is quite slow for alginate. But overall, if we say yes, it biodegrades through hydrolysis. Then chitosan is degraded by the lysozyme enzymes that are there in our cells. So you know about lysosome, that has the enzyme called lysozymes. So those enzymes, so the action of those enzymes, the chitosan is broken down and degraded. Like similar to alginate, dextran, which is mainly made up of glucose, right? So, and glucose, you know, is a dietary component of us. So we eat glucose to get energy. So it's very easy to find the enzymes that are there to metabolize glucose. So it's in using the similar pathways, dextran can get metabolized or it can also get degraded like alginate through hydrolysis. So, means hydrolysis and enzymatic factors both either can play a role in dextran biodegradation. Okay, so I hope it's clear why cellulose is not biodegradable, why other materials that are listed here they are biodegradable in nature. Is it clear? It's clear, right? Yeah. This is the next question.
We've had a problem. Please try again.
This is the next question. Have a look at your screens. So which of the following is a biodegradable synthetic polymer?
B, ma'am.
B, polyvinyl alcohol. Um, so it makes pretty much sense. All the other options are like genepen is a crosslinker, titanium is a metal, alginate is a biodegradable natural polymer. So yeah, B. So these kind of simple questions are also there in your exams or in your assignments. So that's why a combination of all. Now, PVA does not support cell adhesion because so PVA stands for polyvinyl alcohol. It's a synthetic polymer. Okay, so PVA polymer is not porous. It has low protein affinity. It lacks required hydrophobicity, both B and C. So you guys know for cell adhesion, we need like presence of some cell adhesion sites, right? And those cell adhesion sites are generally found in our body's proteins like collagen, fibronectin, laminin, and all, etc. Okay. So when those proteins are there, then only cells will adhere well. So that's the hint I'll give for now. Now you try to answer.
Both B and C.
Both B and C. Okay. So anyone else would like to try? One person is saying it is both B and C. Others agree, or you have any other question, any other option in mind? Why PVA will not support cell adhesion? Which option seems correct to you for this particular question? Option C. Okay. Anyone else thinks otherwise? So we got C and D. What about A and B? Okay. So the correct answer here is actually option D, both B and C. So, sir, I think didn't go too far about surface properties of biomaterials and all. But briefly, I'll explain. I did explain in last week as well, but here also I'll explain. So any synthetic polymer, usually synthetic polymers, they do not have the cell adhesion sites by itself. So we can either graft those cell adhesion sites, like RGD sites, we can graft it through chemical formation of chemical bonds, or we make sure that the surface made by the synthetic polymers, it is of desired wettability. Means it should have desired level of hydrophobic nature or hydrophilic nature. So PVA is a very hydrophilic polymer because of the presence of OH groups. So what happens due to that? When it is implanted in an aqueous environment like inside our body, the water molecules, they are very strongly bound to the PVA surface, because of which any nearby proteins, a plasma protein, okay, that is readily available. So and those proteins, they cannot interact with the PVA surface because the water molecules are there, right? And since the binding force between the water molecules and the PVA is so strong that any competing forces cannot be incorporated, like proteins, surrounding proteins, they cannot compete with that force. because of which the proteins, they cannot adhere on a highly hydrophilic surface. But if it is towards hydrophobic nature, okay, let's say of PCl, polycaprolactone, so that is hydrophobic in nature, but it should not it but it is not very hydrophobic, like super hydrophobicity is also there. Its hydrophobicity is like less than that level. So what happens in hydrophobic surfaces, the water molecules initially does come interact, but this interaction between the water molecules and the hydrophobic surface is not as strong as the hydrogen bonds. It is called hydrophobic interactions goes on. So it is comparatively quite weak. So if a competing molecule like a protein molecule from plasma comes and try to say like, okay, now move on, I am here to sit here. So then it is possible. In that case, it is possible because the forces previously that was there between the water and the hydrophobic surface, it was much more less. So now the protein molecule, they can now interact with the surface of the polycaprolactone or PCL, and it can like interaction can be in two ways when you are talking about protein and surface interaction. If the protein uh, groups or the or functional groups on the protein and the surface of the polymer is too strong, then what can happen? The protein structure can degrade. All right. Or if there is affinity of optimum level, then the structure of the protein can stay intact. So for proper cellular interaction between a synthetic polymer and a cell, what should happen? The hydrophobicity should be such that it should allow addition of protein in its in native state, intact state. It should not degrade. When that happens, when intact proteins interact with the hydrophobic surface, then only the cells can bind to it. So cells are not directly binding with the polymer, rather they're interacting with a very thin layer of or a monolayer of adhered protein molecules. Okay, because these protein molecules are now showing the cell adhesion sites for the cells to adhere. Okay, so it's like so this is like very briefly I tried to explain how synthetic polymers to promote cell adhesion on its own. So a general statement I can say here is if the surface is very hydrophilic in nature, then cell adhesion won't be possible. Also, if the surface is very, very hydrophobic in nature, then also cell adhesion is not possible. We try to balance these two thing in like, and we try to achieve a optimum level of hydrophobicity or hydrophilicity to achieve cell adhesion. Okay. So I hope it's clear to you now why option B and why option D is correct. Yes, no?
Yes, ma'am.
Yes, ma'am.
Okay. Yeah. Now, which of the following scaffold fabrication techniques cannot give rise to scaffolds with interconnected pores? So we are talking about making a porous scaffold so that, and also the pores should be interconnected in nature. I told you why. So that the cells can infiltrate better, better diffusion of media and oxygen is possible. Now, these are the scaffold fabrication techniques that are generally used. Briefly, let me explain one by one if you are not familiar. Electrospinning, here what we do, we take a viscous polymer solution, all right, and we load it into a syringe. And then it is connected to a metallic needle. We apply a electric field so that the and we exclude it using a syringe pump. We exclude the polymer solution out of the needle in presence of a electric field. So what this electric field does, it provides charge to the extruding out drops of polymer. Some charge gets built up, basically a envelope of charge. It can be positive or negative depending on what like polarity is there. Okay, and on just opposite to it, there is a surface. It can be a drum, it can be a plate, and it is of opposite charge nature. So what happens? due to charge interaction, the polymer, viscous polymer, it turns its morphology from a drop to a cone type of shape, and fibers starts coming out of like very thin fibers starts coming out, and it deposits on the oppositely charged surface that is just opposite to it. So the polymer itself, during the process, whatever solvent is there that you have used to make the polymer solution, that solvent evaporates and you get solid fibers which you can hold. All right. So that is how you create fibrous mat, and that fibers can be of varying dimensions like nanometers to micrometers. So you have to optimize your parameters, electrospinning parameters, in order to get fibers of desired diameter, and ultimately you will be able to use it for tissue engineering application. This is called electrospinning. In presence of some electric field of some magnitude, you are creating fibers. That's it. Then 3D printing, there are various types of 3D printing. General approach is what we do in 3D printing. We make ink, or we create a bath of ink, and the material gets deposited in a layer by layer fashion, either from uh, uh, down to top, like the surface is there and the material gets deposited in a layer by layer fashion, and you give a structure like this one that is upright, or you can get a structure like this ulta as well. Okay, so that bottommost layer gets formed first, and then as the stage moves up, the subsequent layers get deposited and you get a structure which is upside down. So that is 3D printing, a layer by layer approach, but the material is deposited in a layer by layer fashion. Okay. Then freeze drying, or it is also known as lyophilization. So here what happens, you first freeze your sample, make whatever water content is there, it turns into ice, and that solid frozen sample is dried under very low vacuum and very low temperature. Okay, so then what happens under these conditions, the process of sublimation starts. Means directly the water molecules, they convert from crystals, ice crystals, to vapors. There is no intermediate liquid phase there. Okay, and this is only possible when we keep the sample at a very low vacuum and very low temperature. This is called freeze drying. And there is another technique called membrane lamination. Here what you do, this is very similar to 3D printing, but here you create sheets. One layer is a sheet, all right? And this sheet, they get a sheet of some polymer. It gets like stacked on top of one another in order to create a 3D scaffold. That is called membrane lamination. Now, now if you have understood what I've just said, or um, watched the lectures on the scaffold fabrication techniques, now you try to answer which of these techniques do you think cannot give rise to interconnected pores, or cannot give rise to a porous scaffold. Option A. Okay. Okay. Anyone else would like to try? Maybe later on after this session, you guys can have a look at videos. So multiple videos are available in YouTube where you can see each of these phenomena, like people are doing and demonstrating. That will give you a better idea about how these techniques work, for what result you get out of it when you use these techniques. So not only the technique that is listed here as an option, but also the techniques that sir discussed in his lectures. All of those techniques, like you can see videos, proper explanatory videos of those techniques demonstrating. So that will give you a much more physical meaning to these terms, right? It will be able to understand why it is used, when to use, what outcome you will get. All right. I'll wait for a few more seconds. Think over it.
Electrospinning.
Electrospinning. Okay, we already got electrospinning. What about 3D printing or freeze drying or memory lamination? Does nobody think that they might be correct? Okay, so the correct answer is membrane lamination. Let me explain why. So, electrospinning, as I told you, you deposit fibers. Now, these fibers are very like individually, if you try to see the fibers to naked eyes, you won't be able to. You need electron microscope in order to see those fibers individually. So, the outcome of electrospinning a mat, is a mat of fibers. You can create multi-layered mat, means multiple layers of fibers are deposited on top of one another, or you can create single layer, so depending on how you optimize your method. And what happens, since the solvent gets evaporated, the polymer is in solid form, so they try to like interlink with one another. Okay, some physical forces act there for the fibers to interlink and then create some empty spaces that forms in between those fibers. So because of why, you can get a porous scaffold if you use the electrospin mat as a scaffold. Okay. Whereas in 3D printing, it is up to us, or up to the person who is doing 3D printing. He or she can add pores of different sizes of different geometry by like making the solid model. So we create a CAD model, and depending on what type of porosity we need, whether we need large pores, small pores, we can 3D print it. So it's actually very easy to create pores of different sizes in 3D printing. Then freeze drying, here what happens? Due to sublimation, just like I talked about, the ice crystals, they sublime directly to vapors. What happens? It leaves behind the empty space. Whatever space that was occupied by water molecules, uh, that like gets empty due to freeze drying. Because of that, freeze drying can also get give rise to a very nicely porous, highly porous scaffolds. Whereas in membrane lamination, I told you we are just making membranes and we are stacking one top on and the other. All right. So here, like you won't be able to get porosity until and unless you do something extra here. Means you can create just simple film, thin film. You cast your polymer onto a mold, and then you cross-link it through some way, and you get a film. And these are one membrane, and then you stack on one another. All right, and stacking, during stacking, you again cross-link the membranes to one another so that it stays intact. And then if you want to give some 3D shape, particular 3D shape, you can carve it out of it. So that is called membrane lamination. You can do porosity here, but you have to add something extra, like some porogen you can add, like salt crystals vagera. So these salt crystals, if you dissolve in your polymer solution before casting membrane, and what you do, you dissolve out the salt crystals after the polymer is cross-linked, then the salt, this pores of the size of the salt crystals get formed. So that is like combining membrane lamination with the technique solvent casting and porogen leaching. So I hope now it's clear why option D is correct. But I strongly urge that you guys see some videos. It is readily available over internet. Demonstration video I'm talking about, where people are demonstrating it. They are doing the method and recording it. So that will give you a much better idea about how these techniques work and when to use it, what outcome to expect. Okay, do this homework. You will understand better if you have not understood now. All right. Now this is next question. Sponge is not a hydrogel because you know sponge, right? That we generally use in our homes to maybe clean our dishes. It does not. Yes, it doesn't. You repeat? Can you repeat?
Option B, option B.
Option B, right. So yeah, it makes sense. I'll not take too much time here in this question. It is correct. Sponge, since it does not hold water, it is not a hydrogel. The unique property of hydrogel is that it can hold water and create a aqueous environment. That is why hydrogel is preferred when you are trying to artificially create a tissue. We are trying to mimic the aqueous environment that is there in our body through the use of hydrogels. Okay. Now, which is not a chemical cross-linking method? So you understand crosslinks, right? You're trying to link adjacent polymer chains with one another. Now you can do it physically or you can do it chemically. So when you're talking about physical bonds, they are reversible in nature. When you're using chemical bonds, you are making permanent covalent bonds through using some reagents, right? So now you answer which of this option is correct for this particular question.
Option B.
Option B. Okay. What about others?
Crafting.
Okay. Crafting. Others? Any other answer from anyone? Okay. So the correct answer here is actually option C, hydrogen bond. So as you guys have definitely heard hydrogen bonds a lot, but let me remind, hydrogen bonds are physical bonds. Here, a hydrogen molecule gets attracted to highly electronegative atoms like oxygen, nitrogen, and it creates a physical bond. It can get easily break, so it is reversible in nature. We just need the presence of a hydrogen in close to a electronegative atom. That is why it is a physical bond. It is not a chemical bond. Whereas grafting is like you are trying to add, say, RGD sites or RGD peptide through chemical bond as a branch to a polymer chain. That is grafting. That is an example of grafting. Means small, short length polymers are covalently linked to long chain polymers. and you're trying to create, so if this is your long chain polymer, you have added small peptides or smaller version of smaller oligotides or something like that, they're added. That is called grafting. Whereas Schiff's base is actually a group of chemicals which are known to uh, involved in Schiff reaction. So it's basically a chemical reaction where alcohol and aldehyde they react. Okay, and it is very commonly used to cross-link um, polymers of different synthetic polymers, right? For some synthetic polymers to get different type of uh, cross-link polymers. So I don't remember what are the shift bases right now, but just have Google it and you will get to know. Okay, whereas disulfide bonds, yes, it's a chemical method where you're trying to create sulfur-sulfur bonds like this. Okay, so yeah, I hope this question is clear to you. Have a look at what are the shift bases that are generally used. Okay, maybe a small homework for you guys. What are shift bases for what type of polymers? It is the most appropriate cross-linking chemical. All right, just have a look briefly. And if you Google it, you will get small answers. Okay, if no further doubts, this is the last question for today. So there is the dry weight and the wet weight of a hydrogel given. You have to calculate the degree of swelling of this hydrogen. So if you know the answer, you do it. All right. Take a pen and paper and you start doing it on your own. Meanwhile, I'll also do it on your screen. For those who are not get to know or like not understanding how to approach. So what is the like how will you calculate degree of swelling? The simple formula is um, wet weight minus dry weight with respect to the dry weight, and it's a percentage quantity. Okay, so we are given the values for dry weight and wet weight, so we have to just uh, put those 150 mg 50 mg divided by 50 mg, okay, into 100. So we'll get the percentage. It's 100 by 50, right? It is 200%. Okay, so degree of swelling in this case is 200%. Okay, understood, everyone?
Yes, ma'am.
So many papers, literatures, if you see that they are trying to calculate the degree of swelling of hydrogens. So generally, we don't do it for one particular time point. We do it for a duration, like for 24 hours, after 48 hours. So we take the hydrogel, we first dry it, maybe through freeze drying or air dry it to remove the entire water content, and then we wait. That is our dry weight, and then we dip it in water or some buffer like PBS, phosphate saline buffer, and we dip it for say 24 hours, for 48 hours, for 72 hours, and then we weigh the wet, and we get the wet weight values. So over a period of time, we get the values of degree of swelling and try to understand if there is a change in swelling. Means we're trying to understand the kinetics of swelling of this hydrogen, because what happens, why it is needed to understand the kinetics, we culture our cells within the hydrogens for a longer period of time, and we want to know how much the swelling property is affected when you are culturing for a long period of time, like say 14 days, 15 days, 20 days, 30 days. Then what happens? Or will the structure undergo degradation? Right? If it starts to degrade, if the structural stability gets compromised, then obviously degree of swelling will reduce. So that is why these kind of studies are done, and it's possible that you might get a similar question where the time is an important factor that will be mentioned to you, like say at 24 hours the degree of swelling was this, at 48 hours it was this, some dry weight value is given, calculate like what was wet weight at 24 hours and then 48 hours. Like similarly you will get like complicated or slightly complicated version to this particular concept. Okay, so this is it from my side for today. Now, I have some time left for Q&A. If you have any questions related to today's session or any lecture content that sir discussed or previous week's content, anything you have in your mind that you want to ask or discuss, now is your time. Were you guys able to understand the questions that I discussed today? Anyone missed any question? Yes, you're saying something? Go ahead.
The session was wonderful.
You understood
Yes,
the
ma'am.
questions? Yeah. All right. Thanks. Um, anyone has any doubt related to any question? Maybe they missed because they were joining in between and they were not able to get the entire gist of that particular question. Do you guys want me to repeat anything? All of you are done with assignment, this week's assignment. Do you have any doubt related to those assignment questions? You guys are watching the lectures or not? Last week I suggested some books related to tissue engineering. If you find any doubt that is not clarified by me or by sir's lectures, maybe those books can be of help. All right. So, yeah. Any queries from anyone? Any comments? Anything that they have in mind that I maybe misspoke about anything, or any feedback for me that they have and they want me to improve on anything? Okay, then, if there are no further queries from your side, uh, we'll wrap up today's session. Thank you all for joining, and the video lectures that is getting recorded, it will be uploaded, and you will find the notes as well, so that you can have a look at it at your leisure times. So I'll see you next week, same time, 4 p.m. on Saturday. Okay. You guys can leave the meeting.
Thank you, ma'am.
Thank you, ma'am.
Thank you for joining.